see my desktop, and yeah. So I will get started to try Chen Kun's Rmarkdown thing as a user. Yeah, just pretend I have no background. I just get started from the link Chen Kun shared in the in the chat or in the slide. Yeah, this is the link, and let's see what happened. Yeah, I just copy and paste the link to my browser, and then you will immediately see the GitHub page. Is everybody on the same page? Can you see the GitHub?
Yes.
Yeah, yep. I guess yes. Yeah, and then if you have already successfully generated that PDF, that HTML, then you are good. And if you encounter any issues, let's go through them one by one. Yeah, step by step. And after you go, arrive this page, you will see a code button here. If you click on this button, there is another sub button, download zip, and then we click on this one. And in my browser, it will download to, yeah, here. Yeah. So they will open this folder automatically. Yeah. In this folder, you can see several files. Like this first two CSV files, one of them are original gene expression data. And the other one, the smaller one, is the metadata. Yeah, showing in each of the cells what's their cluster, what's their time point, and whether it's a control disease. So the format is something like this. Yeah, and we also have other several things. For example, the .rmd files. They are this coding part. Yeah, Chen Kun just showed. And this one is used to generate this HTML format. Yeah, just double click on this button. Yeah, and as you can see, this is a report you can generate directly from Rmarkdown. Yeah, I will not go back to the coding part, but I'm gonna show what we're gonna generate here. Yeah. So first of all, how to do the things I just did. Yeah, you can click on this. This is the same link I just used, and then download the zip code and unzip files, and you will see, the files here. For each of them, there is a very short description, and then in step 3, you can change the working direction, because this one is Chen Kun's computer. If you want to modify that RMD file, you need to change the direction exactly to the folder. Yeah, where you located this folder, I downloaded. Yeah, and then this is the thing we generated from that RMD file. We can open this sub-menus. First of all, the data. Since it's a large data, we just directly go to 3.2. Yeah, this is how you load data in Seurat. Yeah, which means the data already, went into Seurat, and then you are ready to use Seurat. And we normalize the data. Yeah, I will touch base why we need to normalize data later. And then we identify highly variable features. So those features are an interim step of Seurat. You can do something, like we use the top 2,000 features. You can use top 1,000, you can use more, you can use less. That's totally adjustable. Yeah, but usually people use the default 2,000. Yeah, scaling the data and perform a linear dimensional reduction, the PCA. Yeah, I think in our previous slide, we mentioned the PCA is kind of linear dimensional reduction method. It's widely used, and it's already assembled in the Seurat package. And then we can determine the dimensionality. That's something like how many PCs you want to use in PCA. And then based on the selected PCs, you can do cell clustering. And then This is the plot, colored. Yeah, so here is a kind of a generated figure. Yeah, and different colors means different time point. And then you can color them using, of course, you can using other metadata, like you can use cell type to recolor those identified cells, cell clusters. You can use time point. You can use control and disease. You can do whatever you want, after the cell clustering. Yeah, like just using the code, showcase here. Yeah, like here. Yeah, that indicate which column in the metadata you will use. Yeah, and then 3.12 is the very popular differentially expressed gene identification. And in this table, like you can change here. Like in this example, we are identifying the genes differentially expressed between control and disease. Absolutely, you can change here to cell type 1 and cell type 2, which means the table will showcase all the genes differentially expressed between cell type 1 and cell type 2, or between 10.1 and 10.2, or between the two groups you prefer to compare. Yeah, and then this is a table you generated. In the HTML format, it's very interactive. You can make re-rank based on the p-value, based on the average log fold change. In this way, you can see the minus log fold change. In this way, you can see the positive log fold change. That's the p-values. Okay, and then you can pick some top differentially expressed genes to plot their variant plot. So which means you can see in this specific gene, they are significantly differentially expressed between control and disease. Yeah, in control this is very high, very active, but in disease it's not. Yeah, we can see some others like this. Yeah, and then the last one is the functional enrichment analysis. And based on this subtypes, we have 4 categories. Because this is the example data, the performance, the p-value is not very significant. Only one of them is like minus less than 0.05. Majority of them make nonsense. Yeah, that's because we only picked 10 genes, I believe. So this usually will not lead to a lot of useful information, but just showcase the results. And you can change different geo categories, CAC pathway, and if you have your own, interested, annotation like regulatory pathway, signaling pathway, you can add, to this analysis. Okay. So basically, this is the HTML format. Yeah, very interactive. You can use it to interpret. Yeah, you can use it. Open this HTML and talk with your collaborator or report to your boss directly. Yeah, it's very convenient. Yeah, and we go back to this PDF. Yeah, and we can open the PDF. Yeah, for example, you are preparing a report of your assignment. You are preparing a paper. Sometimes people prefer the PDF file, and then you will see this is what the PDF looks like. Basically, same thing. And but in a PDF version. In PDF, the disadvantage is we cannot have those interactivity. Means the table are dead. That's static. Yeah, you cannot adjust the table rank. You cannot adjust other things. But the PDF is very stable version. Yeah, and see basically this is how you scale the data, how you perform the PCA. The previous page, how you normalize the data. So basically, this is the information you can put in the method section of your paper, right? And then when we come to some figures, maybe this figure is not directly answering your question. Maybe this figure can be moved into your supplementary file. And here, maybe this is a very interesting, results. You can directly put it into your main body of the manuscript. Yeah, and So basically, that's the same thing like this figure. Yeah, when we talk about the table here, it's not active, interactive. That is a static table. Yeah. Okay, so this is the PDF version. So as a user, I have gone through the whole process, how you use this link. And on your computer, if you want to change the code, you can definitely do some cooking there and change the data size to your data sets in the project you are working on, and then you should be able to generate that quality of report, yeah, with all the description, coding part, and the results together in one, yeah. I personally encourage every single student, either GRA or undergraduate student in my lab to use this when they do their project. Yeah, when they work on the project, sometimes it's hard to track everything. Yeah, maybe after you get some results very interesting, but after 2 weeks, yeah, you cannot find it. Yeah, and you cannot repeat it. You cannot reproduce it. That's always a problem. Yeah. But with this one, we just accumulate our analysis and just accumulate the new insights within one file. Every time we just go through this one, and that's very, very effective. Yeah. So I as a user, I'm just testing the whole thing, and I'm stopping here for questions. Any question in using Rmarkdown, in repeating what I just showed. So please feel free to ask. Both Cankun and I are here to answer questions. Is there any? Okay. It seems everybody can access this code and file, and hope you enjoy the part. Yeah. And this is not only specifically for single-cell RNA-seq data analysis. You can definitely generate other pipelines for your data analysis. For example, I'm working on biological network construction. You can definitely use the technology, yeah, and use the template to do your own pipeline. Yeah, that's definitely something deserve your effort. Okay.
I don't really have a question, but I just wanted to say thank you because I use Rmarkdown a lot, and I really gained a lot of tips from watching you and your coworkers showing what you do. So I wanted to say thank you very much.
Okay. Yeah, I appreciate. Yeah, I appreciate the good words. We just share what we have here. Yeah, and I just share something I think will benefit you or your lab even. So that's our purpose. Yeah.
Yeah. Thank you so much.
Thank you. So, Narun, can I just move forward?
Yes, I think it's a good time to move forward.
Okay. So, just a quick review of the other kind of analysis technique. So we introduced the Seurat. We introduced the Rmarkdown to build a pipeline using Seurat, and then the other part, computational or enabling technique in bioinformatics is web servers, or web databases. So this is in-house web server, so-called IRIS-EDA. Yeah. So this one will be introduced next tomorrow, and also by Cankun, I think. Yeah.
Yeah. We can hear the video. Sorry.
So anybody? Yeah.
So talking about, can you reshare the slide and
you are not okay.
there's a button and the share system volume when you enable sharing.
Oh, you cannot hear the voice, right? From the video?
Yeah. And can you reshare it?
How about now?
I cannot hear it. So I think you need to stop sharing and reshare it. So when you start sharing, there is a button. You need to click on share system, sound or volume.
No, I can only see share my desk, Windows, or the slide. So Sankun, can you see my slide, but you cannot hear the voice from the video, right?
Yeah, I can share this slide.
Okay, good.
If I may interject, sound is a pretty typical issue that we have in these meetings. It might be better if you just share the link and we watch the video on our own and come back.
Okay, that's good enough. Yeah, I will take
This
that.
is the.
Oh, if that just worked, then never mind.
Okay. Just go ahead. Yeah,
thanks.
IRIS-EDA web server, the integrated RNA-Seq data analysis and interpretation system. So the first place you'd want to go here on the server is the Submit and QC tab. This is where you would submit your data, or you can work with one of the example data sets we have provided. You can either work with one of the two bulk RNA-Seq data sets, or you can go with the single-cell RNA-Seq data, or you can also load your own data using one of these options. If you load your own data, you'll have a submit option for the count data, which would be raw count data, and also the metadata, so the condition information. For this tutorial, we'll just stick with the small example data set. The filter cutoff is going to be filtering out any rows that have a count data sum less than this specific number. The default is currently set to 10, and transformations for counts, just for summarizing the data and displaying it. There's a few different options here, including no transformations at all. The default is the normal log transformation. Once you have your data uploaded or selected the example data you'd like, you click the submit button, and that populates the data into the server. The first piece of information, the first couple of things that pop up is actually a view of the data itself. The first part is actually the expression data with the gene IDs on the left, and across the top, the sample information, and these are the raw read counts. The sample metadata just shows you what your condition information looks like. So here we only have 2 different conditions across 7 different samples. And we also get a little bit of information related to the number of IDs, so the number of genes that were present, pre-filtration and post-filtration, just to get an idea of, based on this filter cutoff, what exactly was done. You can also move on to the count summary, which sometimes, depending on your data size, will take a little bit of time to populate. It just gives a brief overview of the data itself. So some box plots related to the counts for each of the samples. All of this is highly interactive. It'll tell you the quartile information, medians, and such. For each of the samples. It also allows you to deselect a couple of options if you're only interested in looking at a few of the options, or if you double click, it selects only that one, and then you can add in a few other ones also. These plots are downloadable either as a PDF or a PNG static plot. The next part in the count summary is the count data distributions. This is the distributions, a histogram for the read counts of each of the samples. It is pretty populated here, but again, it's interactive, and so we can deselect some of the samples and scroll over to get some more information. Again, double click and we can select fewer if we would like. This also has the property that we can select only certain sections of the graph. Double clicking will allow us to back out to the default picture. The last piece of information we have here is total reads for each of the samples. Again, interactive to some degree, and you can select particular samples you're interested in, double clicking to back out of all the. We can also download those as static images as well.
Cool. Thank you, Sankun. Yeah. Yeah. I just showcased the web server. The voice, the voice was from my previous postdoc, who is currently assistant professor in University of South Dakota. And he developed this server, and currently this server was maintained, how to go to next, currently the server is maintained by Tan Kun. So besides those initial introduction of the server, we will go into more details regarding how we can do other data analysis, like a hypothesis-driven differential gene expression analysis, or discovery-driven gene cluster, cell cluster, or PSA analysis. Eventually, we can support data submission, both of the raw data and the results, to the GEO database, to support the FAIR principle. So that's what I mentioned. The other kind of technology. For some students and researchers who have sufficient coding experience, you definitely like to use like R package. You can play with that, But for some other biologists, and it's really still a need, to free their burden of programming, Then that's why people need web servers like this in this public domain. Then you can really talk with your computer. You just let them know, I need more, more gene expression, more differential expressed genes, and then you can just adjust the cutoff on the server, and immediately you will get updated gene list. So more interactivity. So that's something definitely we will introduce tomorrow. And okay, today the rest of the, I think I still have about 35 minutes. I guess 30 minutes, and then I will leave some time for questions. So I'm introducing some single-cell RNA-seq data analysis pitfalls. So compared to giving you a list of do's, I prefer to give you a list of do nots. So let's see what kind of pitfalls you might encounter in the future. And if you already know the strategy to take with take care of those pitfalls, then I think your analysis will be good. So first one, priority doesn't equal to high performance. That's happened when you want to pick a software. I just give you an example. So here we are talking about the reads alignment. That's the very first step when you get the sequencing data, no matter bulk or single cell. You have to map those reads back to a reference genome. Let's say human genome. So in that step, multiple tools are available in the public domain. I do believe some of you should have experience in analyzing in reads alignment. So please just let me know, by saying yes when you see a name which is familiar to you, in my next slide. Like the first tool is a map spliced tool. So anybody heard about that or use it? Okay, next one, CRAC, GSNAP, GSNAP. Nova Align. No. So Top Hat. So does anybody heard about Top Hat? I think this is a top popular one. Developed by a group from Johns Hopkins. So there's a lot of citations. But based on this Nature Method Review paper, published in 2017. Top Hat has lowest race mapping performance in terms of a large-scale simulation analysis. So that's the point I mentioned. Popularity doesn't equal to high performance. When you want to carry out analysis, especially you are not very familiar with, the good way is to check whether there is already a very good review paper published in a reputed journal, and then based on the performance driven recommendation, you can select 2. That will help a lot. Yeah, not only like that paper use this tool, I will use it. Then there's not a very good rationale. Okay, second example, also Pitfall I, popularity doesn't equal to high performance. Now we are talking about analysis specifically for single-cell RNA-seq data. It's so-called single-cell trajectory inference. Yeah. So based on the graph methods, yeah, that's showcased here. Yeah, there are different kinds of trajectory analysis. Using this trajectory analysis, you can infer the cell development, cell differentiation, yeah, in a specific context, like in cancer, yeah, like in Alzheimer's disease, like in a normal tissue, yeah. So anyway, there are a lot of tools, yeah. In this Nature Biotechnology paper published, I think last year, they reviewed 41. Actually, this is more than 70 trajectory tools, but they selected 41 tools, yeah, and then take a very thorough review, yeah, and then the performance list is here, and in which Monaco, the rank the 21st, Monaco is I think it's the one most popular, yeah, with a high citation, but actually their performance is only ranked 21st, yeah. So this is another example, yeah. So so that's my suggestion to all the students, yeah. When you do your analysis, yeah, you really follow the recommendation based on performance, not only based on citation or based on other papers. Okay. Okay. Second pitfall, so-called dropouts. Yeah. Not sure whether Anjun is here. Yeah. Actually, he helped to prepare some slides here. Maybe he has more input, but dropouts is an issue caused by the sequencing technology, which means the gene may have a very low expression value. When that happens, the technology cannot capture the low expression value. So they just take a 0 there for this gene in this cell, which means some zeros are not true. They should have a original value, even that's low. So people call those zeros should not be 0, a dropout. Yeah. And Anjun, are you there?
Yeah.
Yeah. Can you go ahead to give us more details regarding this?
Yeah, sure. So I think Dr. Ma just mentioned the dropout issue, and which is a very critical issue for single-cell data analysis, not only for the single-cell RNA-seq, but also in single-cell ATAC and single-cell like DNA-seq methylation. So the reason for that is we need to focus only for the one cell, and which limited the ability to measure the true value of how a gene expressed compared to the bulk RNA-seq data, where we have a lot of cells and a bunch of expressed genes. So the possible explanation listed, there are two. Dr. Ma just mentioned one, which is the second low resolution, the true expression level lower than the resolution that the machine can detect, then it just turn it to 0. The other one is the first one is like a technical error, where the non-expression will be identified as 0, even it passed the threshold, the machine threshold. Sometimes we also call it like a true, like the mean value could be 7, and then you have a gene expressed as 8, but there just an error occurred, and the machine missed that, so still give a 0. So these two forms the dropout event. And there are also some true expressions. So the hard way here is how to identify the true zeros and the false zeros. If we can identify the true zeros, then the problem solved. So the way to statistically or computationally solve the dropout issue is called imputation. So probably Dr. Ma, you can switch to the next slides. Oh yeah, So imputation, there are just like clustering and trajectory, there are a lot of tools, computational tools in the public domain that try to solve the issue. And there's I remember there are 2 existing review papers that discuss such a problem. But here I showed you the newest one, which have not been published in the journal, but only on bioRxiv, that evaluate 18 imputation tools with 11 evaluation criteria, such like a benchwork. So among all these imputation tools, there are 3 in the MAGIC, Kin, smoothing, and Seurat. These 3 are found to outperform all the other methods constantly. So the constantly here means like for both SMART-Seq and text genomic data, all of them show the best performance than the others. So I want to mention the MAGIC here. The MAGIC is, I think for imputation, it's the most popular one. It published in 2017 on Nature Method, on Cell, and it using the smoothing method. But one issue it introduced, and actually for all of the imputation methods, is that they will introduce the false positive signals, which means actually they cannot truly detect all the true zeros and only recover the false zero values. Okay, so those we call the false positives, and such false positive zero, when we change it to the some numbers, it will introduce more the bias and may cause problem to the following analysis. Yeah, and one thing here, because the smoothing method is considering only the neighbor cells and the genes, their expression levels of the neighboring cells and the genes. And so it just like a normalization for each of the expression value. So that's one of the reasons why it has a higher false positive ratio than other methods. So so considering that the dropout issue is still an ongoing problem that need to be solved, so we are now developing a new computational method that using the graph neural network to model the single-cell RNA-seq data. So here we use a strategy that consider the cell type or the cell clusters into the imputation. So here the hypothesis is that we consider the imputation should be like performed both inside of the cell cluster and for the overall cells. For other methods, imputation method, they do not consider the cell type. Okay, and also the second one is we are the first group that using the graph neural network in performing imputation, because we can consider all the cells as a graph, and the graph neural network is one of the best way to solve the graph problem. Okay. So here just a brief introduction of our framework. So we have the row expression matrix, and we do 2 ways. One is to go to the LTMG model to have the regulatory signals. And then the regulatory signal matrix will be utilized as a regularizer, okay, to send into one of the autoencoder. We call it feature autoencoder. Okay. So the feature autoencoder will give us a kin graph. And then the graph will send into a graph autoencoder, which includes a graph neural network, and give the cell embeddings. From the cell embeddings, we can do the clustering to get the cell type, and the cell type information will then send to the imputation autoencoder, which are formed by multiple similar feature autoencoders, as we just mentioned. And each one will only consider the cells within a cell cluster. And then the output will be combined as an imputed expression matrix, and then sent back to the feature autoencoder to complete the circle. Okay. And this iterative process will be terminated once the cell clustering converged compared to the previous circle step. And then we output both the cell clusters and imputation matrix. So we have some preliminary results that showing both the imputation and the clustering from our framework. So on the left side, the top one, you can see we compile the raw data. Here is just the two gene. I didn't show the gene names here, but the X and Y are the expression of two genes, and these two genes should be have some correlations in their original paper. Okay, so using the raw data, we can hardly find any patterns or correlations. But after SAGN imputation, we can find a two-way correlation that separate by the cell clusters. And actually, these are also like a part of trajectory part. Okay. And down here is the AI results evaluating the cell clustering, how similar our cell clustering results compare to the original benchmark cell types. And the red one is our SAGN results. Very close to 1 means almost 100% the same, and competing to other 7 existing imputation methods. Okay, so our results showcased the SAGN framework can accurately capture the gene pair correlations and enhance their signals, and also can help the cell clustering results.
Okay, thank you, Andrew. Yeah, so this slide is showing some ongoing projects here. Andrew is leading this deep learning based project for gene imputation. So we'll go back here, showing this triangle. Yeah, we missed. So the sparsity of the single cell data are mainly induced by this dropout event, and mixed together with those true zeros, those dropout issues will make the data analysis and the results interpretation very, very challenging. And that's why there are so many tools that developed dedicated, in gene reputation. Yeah, basically they want to recover those genes from 0 to some non-zero values. Yeah. And our work, so-called SAGN, is short for single cell graph neural network. Actually, I want to mention that the deep learning is also a very important direction within the lab. Yeah, within my lab, we are trying to formulate some like single cell related projects into the machine learning, deep learning framework. Yeah, I know some students from computer science department. If you're interested in some deep learning application in single cell data, yeah, you can feel free to contact us. Yeah, we have a lot of things to share. Yeah, okay. That's the imputation. I think that's our second pitfall. The third pitfall is batch effect and normalization. I think during Chen Kun's pipeline, yeah, you can see before we rarely do cell clustering, we rarely do differential gene expression analysis. We need to do imputation and we need to do normalization. Yeah, so normalizing single-cell RNA sequencing data. Yeah, this is also a Nature methods paper published 2 years ago. Yeah, they summarize, they compared, summarized the different normalization ways for single-cell RNA-seq data. Yeah, so why we need normalization? Yeah, because the main reason, one of the main reasons is batch effect. Yeah, so yeah, so we need to remove those batch effect, especially in the single-cell level. Yeah, so Chen Kun, are you there?
Yeah.
Yeah. So I do believe this slide is prepared by you, right? Yeah, you can give us more details regarding the batch effect. Yeah.
Yeah. So when we talk about batch effect, so the problem usually occurs when we want to integrate data sets from multiple sequencing output. So if we look at this figure, so we have the sequenced data from the cells that added drug and the cells without the drug. If we just simply combine these two data sets, you can find that the cell t-SNE, the dimensional reduction results, that these unaligned data sets are most dominated by the drug effect. So you can see that the cells and the drugs added drugs tend to clustering together. However, when we are doing single-cell analysis clustering, we want to see the cell types, that similar cell types are clustered together. So that's why we need to perform the data integration instead of simply combine the data. So if you look at the image below, after we do the data integration, you can see this drug effect is reduced, and the similar clusters usually represent different clusters represent different cell types. So the reason why there batch effect, I list some example here, like the days of sequencing, the people who done the sequencing, or the sequencing machines like the flow cells, plates, the chemistry, that or the protocols, or the read length, all of these can have effect on the cause the batch effect.
Yeah. I have one more to add. So different sequencing technologies, like in single cell, we mentioned there is SmartSeq2 and there is 10x genomics. If some of you can recall that slide I showed, there's a comparison between SmartSeq2 and 10x genomics. In SmartSeq2, it's a full-length mRNA sequencing technology, so we can get more genes sequenced, identified, but in limited number of cells, versus to the 10x genomics, we usually get very limited number of genes identified, but we can sequence way more cells than in SmartSeq2. So different technologies will generate totally different, different types of data. Yeah, both of them are single-cell RNA-seq. So if you are analyzing or integrating SmartSeq2 and 10x genomics RNA-seq data without normalization, I bet the results are very hard to interpret. Yeah. So beyond that, like Chen Kun listed here, there are other 5 bullets here, all causing batch effects. So we need to do the normalization. Yeah. So Chen Kun, do you want to say something for this slide?
This page is from a review paper about the benchmark of batch effect correction methods published earlier this year in Genome Biology. So this figure shows the overall performance of all of these software on these data sets. So overall, the Seurat 3, Liger, Harmony are recommended in this paper.
Okay. Yeah. So thank you, Sang-Kun. Basically, we showcased the normalization, and we showcased the batch effect as Pitfall. And here is a recommendation, not by us, by this genome biology paper published early this year. And the 3 recommended packages are listed here, including Seurat. Yeah. So that's the reason I mentioned previously. If you only have limited time to focus on this learning, so get started with Seurat, it will benefit a lot. Okay. Yeah, we're gonna go to the next one. Next one is kind of simple. Yeah, the Pitfall is using t-test for differential gene expression analysis. Yeah, differential gene expression analysis is not new, a long time. Yeah, maybe 2 decades. So we have the gene expression analysis, and we want to know whether this gene is differentially expressed between 2 groups. Yeah, maybe treatment and control. Yeah, maybe different cell types. Yeah, or 2 groups you are interested in. Yeah. And there are a lot of Nature Science papers using t-test for differential gene expression analysis. I do believe not everybody here know t-test exactly, but basically t-test is to identify some genes here which have a significantly different pattern in group 1 and group 2. So that's why people use it for differential gene expression analysis. But I listed as a pitfall. Yeah, even a lot of reputed journal published a paper using t-test. Yeah, because all those statistical analysis usually have a hypothesis, underlying hypothesis. Like using t-test, the p-value minus 0.01, then this gene can be identified as a differentially expressed gene. So what's the problem? Yeah, the problem is, not showing here. The problem is t-test, the underlying hypothesis of t-test is both of the red group and the samples in red group and in blue group, the samples should follow normal distribution. Yeah. Otherwise, t-test will have a biased p-value generated. So you said why we cannot claim a set of samples follow normal distribution. So the thing is, usually we have a very limited number of samples. Not in the single cell, data. In the previous, like we call small big data. If you are comparing 10 samples amongst another 10 samples, you never use t-test because you can never claim 10 values follow a normal distribution. You at least have 30 something. Yeah, but nobody, really understand. Not everybody really understand the underlying hypothesis of t-test. So I saw those papers. They use 5 versus 5 comparison, and then still use t-test, and then they still publish very good papers. So that's a pitfall we should solve. And actually, in statistics, there is a kind of test. Generally, the category is so-called non-parametric test, which means we do not, based on any hypothesis, any distribution, and we do not have assumption that your data follow normal distribution in disease and follow another normal distribution in normal tissues. Yeah. So in that case, yeah, for example, Wilcoxon test. Yeah, Wilcoxon test is the non-parametric test. They only based on the rank of the values in your data in the 2 groups you want to compare. Yeah, And the Wilcoxon test has better performance than t-test. This is one sentence of the conclusion in this bioinformatics paper, which published almost 20 years ago. And but even now, yeah, still people are using t-test, yeah, when they have a limited number of samples. So that's something I want to showcase here, and hopefully you can use Wilcoxon test in the future, yeah, if you are not sure whether your data follows normal distribution. So I just said this is not the case in single cell. Single cell, we have a sufficient number of cells. Yeah, in one cell type, we have 1,000 cells, and in another cell type, we have another 1,000 cells. We are definitely, do not limited by the sample size. Yes, but in single cell level, the things are even complicated. One, due to the high proportion of zeros, yeah, dropout issues, and the other side is in single cell, even you have a lot of non-zeroed genes, the values still do not follow normal distribution. Regarding what kind of distribution it should follow, somebody say negative binomial, yeah, which can be composed by gamma and Poisson. Yeah. Somebody say it's a mixture of Gaussian distribution. Yeah. So we will try to touch base with this in our following slides. But here we just throw out this idea. When you use some statistical analysis, try to understand their underlying distribution, and then you will have a smart choice. Okay. And this is the same one. In single cell level, I list several differential gene expression analysis tools, including B-seq2. Yeah, B-seq2 is the one developed for bulk data. Yeah, but it can be used in single cell data. Yeah, and MAST and SC2P. Yeah, there are several things. And this is a paper, also recent paper last year, focusing on handling zero counts or multimodality based things in single cell RNA-seq data analysis. Yeah, differential gene expression analysis. Yeah, somebody interested in which two have the best performance, you can check that paper. Yeah, and otherwise you can just follow the recommendations on the slides. Okay. Yeah, I want to mention this is still a challenge. This is still an open question. Yeah, open project. Yeah, if you are very interested in how should we confidently identify those differential expressed genes in single cell data, this is a really good research project. Yeah. Okay. We still have other pitfalls. Yeah, we still have other pitfalls, other challenges, and I do believe I will introduce them all tomorrow. Yeah, and I will stop here listening to the group. Any comments, questions? So feel free to ask. Yeah, you can ask questions regarding Rmarkdown we introduced early today. You can discuss any pitfalls I just showcased here. Anything welcome.
As for tomorrow, you're going to be showing some code, correct?
It's code of what?
For what we have going on for this.
Yeah, tomorrow. Yeah, so I think we have shared the Seurat code, we have shared the Rmarkdown code. Those are code we already shared. And we will not share any code for those pitfalls, because the pitfall, we just let you know, you should take those pitfalls into your consideration. Like when you build your pipeline using Rmarkdown, you should keep in mind, hey, this is single cell data. I should do normalization for batch effect. And then I should do imputation for the dropout issues. Regarding which tool you should use, you can check those names on my slides. And I think that's what I mean.
Okay.
Yeah. Tomorrow is code free. Yeah, tomorrow we will introduce web servers. Yeah, we will introduce how you just click on several buttons and then you can generate some beautiful results. That's tomorrow's focus. Okay. Yeah, and during the time people think about their questions, and Naren, I'm sharing the material with you after this this meeting. I will share the slides and I will share the Tankun's Rmarkdown folder, and I will also share the Seurat example data from Yuzhou. And I hope just in case some students cannot get them from online, they can use the local version. How about that, Naren?
She may have stepped away, but I'll get that relayed.
Mm-hmm.
Okay. If there is any more questions, I'll stop recording and see you tomorrow.
Yeah. If no more questions, I'm gonna stop sharing and see you all tomorrow.
Thank you. Good day.
Thank you.
Thank you.
Thank you.
Yeah.
Thank you.
Thank you. Yeah.